Herpes simplex virus.
Herpes was first formally recognized by Hippocrates, who wrote about the symptoms of herpes lesions. A Roman physician named Cellus developed a treatment method for herpes that involved cauterizing open herpes lesions with a hot iron. Herpes is most commonly found in Eastern Africa, with a higher affinity for African Americans than any other races. Around 70% of American adults have oral herpes. An estimated 25% of American adults have genital herpes. 85% of the affected population do not even know they have it because they think it's just a cold sore or the virus is dormant. 67% of the global population has HSV-1, according to the World Health Organization.
Most of the time when herpes simplex virus or HSV infects a person, there are no symptoms. In fact, it usually moves from one person to another in the absence of symptoms. So therefore, it can move through a population silently. Once in a while, though, it can cause symptoms, and typically those are in the form of skin and mucous membrane lesions, which can be divided into infections above the waist, and mostly involving the mouth and the tongue, and those below the waist involving the genitals. There are two types of herpes simplex viruses, HSV-1 and HSV-2, both of which are part of a larger family of enveloped double-stranded DNA viruses, the herpes viridae family. Generally speaking, HSV-1 tend to have symptoms above the waist, and HSV-2 tend to cause infections below the waist. But there's a lot of crossover because both viruses can cause both types of infections. Although herpes is most contagious when there are virus-filled lesions present, it can also spread by asymptomatic shedding, which means that herpes virus can be in saliva or genital secretions, even when there are no signs of a cold sore or genital lesion. Typically, when herpes virus lands on a new host, in other words, a person that's never had herpes before, it dives into the small cracks of the skin or mucosa and binds to epithelial cells receptors, which trigger those cells to internalize the virus. Once inside, the virus starts up the Lytic cycle, which is where its DNA gets transcribed and translated by cellular enzymes, which help form viral proteins, which are packaged into new herpes viruses, which can leave to go off and infect neighboring epithelial cells. HSV-1 and HSV-2 also infect nearby sensory neurons and travel up their axons to the neuron cell body to start up the latent cycle. The sensory neurons of the face have their cell bodies in the trigeminal ganglia, and those around the genitalia are located in the sacral ganglia. So that's ultimately where the herpes virus settles in, for life. You see, the sensory neurons aren't destroyed. Instead, they become permanent homes for the herpes virus. And from time to time, the herpes virus makes a few viral copies of itself and sends those copies back down the axon so they can get released and infect epithelial cells. Since the trigeminal sacral nuclei serve as one side of the face or body, herpes vesicles and ulcers develop on the ipsilateral side or the same side of the affected nuclei. This can happen over and over throughout a person's lifetime with classic triggers such as stress, skin damage, and viral illnesses. Recurrent episodes are usually less severe than the primary infection, and sometimes it has no symptoms at all. When there are symptoms, there might be a characteristic tingling or burning sensation called a prodrome, 1 or 2 days before the blisters appear. In oral and genital herpes, the primary infection is most often asymptomatic. Having said that, in oral herpes, when it does cause symptoms, it usually affects children and it causes lesions around the palate, gum, tongue, lip, and facial areas, as well as a fever and enlarged lymph nodes. The lesions themselves are typically clusters of small, painful, fluid-filled blisters that ooze and ulcerate and eventually heal over a few weeks. In older children and adults, a common symptom is pharyngitis. Most of the time, like primary infections, reactivation doesn't cause any symptoms, but when it does, the most common factor is having a handful of blisters at the vermilion border or the border of the lip on one side of the face. These blisters typically are smaller and heal all over a week. With genital herpes, the primary infection can cause symptoms like ulcers and pustules, which form on the labia majora, labia minora, mons pubis, vaginal mucosa, and cervix in women, and on the shaft of penis in men. Like oral herpes, most of the time reactivation doesn't cause any symptoms, but when it does, the most common pattern is to have a few blisters that resolve rapidly in about a week.
HSV can cause unique symptoms in immune-compromised individuals who tend to have more frequent reactivation and a higher range of symptoms like lesions in the lungs and esophagus. Herpes can usually be diagnosed based on how the skin or mucous membrane lesions look and can be confirmed with tests looking for viral DNA, like polymerase chain reaction, which is the antibody response to the virus, or by growing the virus within a viral culture. Although infections typically resolve without treatment in a couple of weeks, there are antiviral drugs like acyclovir, famciclovir, and valacyclovir that can be used typically or systematically to reduce pain or speed up healing. For recurring episodes, these treatments usually work best if taken when the prodrome starts. This is when before the blisters develop, and a high-dose intravenous antivirals may be given in more severe or life-threatening situations. The herpes simplex virus has a large double-stranded DNA genome that carries the genetic information for the synthesis of over 100 polypeptides. A protein capsid surrounds the DNA and a phospholipid membrane surrounds the capsid. The virus has elaborate glycoprotein spikes that allows it to recognize, bind to, and fuse with few cell types of the body. The viral envelope that becomes incorporated into the host enzyme in the nucleus capsid releases the DNA into the nucleus. In the nucleus, the DNA circulates, and host enzymes begin to transcribe the first set of viral genes known as the early genes. The early mRNA molecules enter the cytoplasm, where they are translated into early proteins. The early proteins, in turn, enter the nucleus, where they're required for viral DNA replication. At this point, the cell begins to transcribe the late mRNA molecules. These mRNA plasmids translate into late proteins. These late proteins include the capsid structural proteins, which enter the nucleus. The late proteins also include the envelope proteins, which are produced in the endoplasmic reticulum and become incorporated into nuclear membrane. The viral DNA continues to replicate. One model postulates that the viral DNA replicates by a rolling circle mechanism. During this latter stage of replication, the end-to-end copies of the viral DNA forms. These copies are cut into individual genomes as they're packaged through capsids. At the nuclear membrane, the capsid and DNA buds off and becomes surrounded by a membrane material with membrane proteins. The virus passes through the endoplasmic reticulum and buds off the membrane and migrates to the plasma membrane. The virus is released from the cell.
Teenagers are most susceptible. In the past, children were regularly exposed to the herpes virus through skin contact with an infected adult or by sharing lip balm and other personal items. This exposure can start the production of antibodies that could come in handy when teens become sexually active and may be exposed to herpes from a partner. But efforts to reduce HSV-1 exposure by discouraging sharing cosmetics along other tactics have been translated to a 23% decrease in HSV-1 antibodies over the past 10 years. In conclusion, herpes simplex virus is very common and there are many drugs to battle the symptoms.